today. Steve Grotsky is a PhD candidate in the Fisheries, Wildlife, and Conservation Biology program at North Carolina State University and a Global Change Fellow with the Department of Interior Southeast Climate Science Center. His research centers on the interface between renewable energy development and wildlife conservation and management. Currently, he is studying the response of birds and invertebrates to woody biomass harvest in the southeastern U.S. One of the goals of his research is to inform sustainable biomass harvesting guidelines specific to the Southeast. Steve earned his Bachelor in Science and Natural Resource Management from Rutgers, the State University of New Jersey, and his Master of Science in Wildlife Ecology from the University of Wisconsin-Madison. Steve is a hard-working guy. He's a very bright guy, and I think has a terrific career ahead of him. So with that, we'll let Steve start.
All right. Thank you, Dennis. That was a very kind introduction. Thank you, Bob. And thank you, everyone else, for attending today. So today I'm going to be talking to you guys a little bit about some preliminary results for the bird portion of my project. And then this webinar will also serve as sort of an introduction to the invertebrate aspects of some of the research that we're doing here. My co-authors are listed here on the title slide. This project, as many of you are aware of, is a collaborative effort between many folks coming from sort of many different angles, so it's an interesting mix. So I'd just like to acknowledge some of those entities as well as some funders, which are listed on the bottom as well. So, what are we doing here today? What are we talking about? So, there's a global energy crisis for which renewable energy might be a possible option to sort of offset some of these issues. So, we know that there are diminishing fossil fuel resources. We have a bunch of people, and all those people use a whole lot of energy, and what we're trying to do here is sort of set the scene where we have a problem. It's an energy crisis, right? So renewable energy is one, like I said, one possible way in which we can help mitigate some of those negative effects of things like fossil fuel consumption, nuclear power generation. It also creates jobs, right? It gives you energy. Possibly mitigate some effects of climate change, but there's sort of at times a darker side to renewable energy. So oftentimes folks think of renewable energy or green energy, environmentally friendly, which in many respects it is, especially in comparison to some of our, some other options, right? Some of those older options, but turns out that there can be some negative effects of renewable energy development on wildlife. So as a wildlife ecologist, I've sort of been charged with trying to get at, obviously renewable energy is a good thing, but we'd also like to get an understanding for how we can implement sustainable renewable energy such that it's not negatively affecting wildlife, or at least that's the goal. So biomass can be converted to energy, right? So taking carbon and turning it into, breaking it down into some other usable forms of carbon that can in turn be used for mainly, for people to use energy, play Xbox, drive cars, whatever. So a lot of this biomass, and what we're really talking about in this presentation, is biomass in the form of harvest residues. A lot of those harvest residues can be found in areas where there is a lot of logging and an established forest industry. So the Southeast, right, the wood basket of the U.S. has a lot of logging, therefore it has a lot of harvest residues, and those harvest residues can be used in a renewable energy context as woody biomass. So moving from renewable energy context to an ecological context, harvest residues are a type of downed wood. So downed wood can be formed in a lot of different ways. You can have senescence of a mature forest, right, with the arrow coming down there, tree fall gaps and whatnot. That's putting down wood on the ground. So also trees can die, which creates a snag, which eventually fall to the ground and becomes down wood in the ecological context. So what we're talking about today is down wood that's created sort of in a quicker way by just forest being, forest stands being cut down, generally clear cutting for timber products, and then what's left is the harvest residue that is essentially, in an ecological context, down wood available to wildlife. So in the case of a clear cut, a young clear cut, down wood, at least early in stand establishment, can provide important structure in terms of cover for wildlife, and then also it can provide some habitat in terms of nesting, also food availability. So in terms of birds can eat seeds that are trapped against down wood in a clear cut. Some birds nest near down wood. So an example would be like turkeys. Provides a little cover and protection for that nest. Also, for smaller passerines, you can see this in your backyard probably. If you got a brush pile, right? Birds fly into a brush pile. They're small. The predator's big. They can't get in, right? The prey, so to speak, is safe there in that brush pile. Downwood also houses invertebrates that breeding birds consume, and then the downwood can also provide structure for behaviors like perching and territorial behaviors as well. So let us not forget that invertebrates also use downwood, and downwood is habitat for invertebrates. So there are many invertebrates that are downwood obligate, which means they spend one or more of their life stages in dead, decaying wood. So an example of that that many people are familiar with would be like termites. Okay, but downwood can also provide structure where predatory ground-dwelling arthropods can find prey. So in many respects, there's similar habitat used by the birds and invertebrates. It's just on very different scales. So knowing that this downwood provides habitat for birds and invertebrates, that begs the question, will removing downwood, an ecological term, will removing downwood through woody biomass harvest, which is a way by which we can get at renewable energy, will doing that comprise bird and invertebrate habitat? So that's our primary question with this project. That this question of wildlife habitat, bird and invertebrate habitat, as well as other wildlife being compromised, has been recognized as a potential, as it could potentially occur. And one of the ways to sort of get at addressing that issue is the establishment of biomass harvesting guidelines. So these are put into place. They're designed to protect, maintain, and enhance biodiversity, site productivity, and then wildlife habitat. So the wildlife habitat portion of these biomass harvesting guidelines are really what this project pertains to. These BHGs have been established in several regions of the United States where woody biomass harvest could potentially occur at an accelerated rate at some point, maybe. And the issue with these BHGs is, they're definitely a great start, and I think one issue though is that they're based on limited empirical data. So the idea is with our project, we can sort of come in and use these BHGs as sort of an experimental framework and then test them and then use empirical data collected from our studies to inform and adapt BHGs further. So in terms of the birds, our objectives are to look at the volume and distribution of retained harvest residue after woody biomass harvest occurs, and then see how wintering and breeding birds respond to those treatments, right, which are representing different volumes and distributions of the harvest residue. And based on that response and the empirical data that we collect, we would like to inform the development of sustainable BHGs into the future. So the objectives for the invertebrates are essentially the same. It's just that our treatments are slightly different, and we're really, with the invertebrates, we're looking at the question at a much smaller scale. As such, the treatments differ slightly such that we're really just looking at the presence or absence of harvest residue and the characteristics of the residue that is retained and relating that to invertebrate abundance in these clear cuts. So just to set the scene here, we're working in North Carolina and Georgia. We're working on the coastal plain of the southeastern United States. We're working on Weyerhaeuser land in North Carolina, and then Plum Creek and Georgia Pacific properties in Georgia. So these are intensively managed pine plantations. All the sites receive mechanical and chemical site preparation, which we'll talk about a little bit more in the discussion. Then they were replanted. Another thing to note is that in terms of site prep, the North Carolina sites were sheared, whereas the Georgia sites were windrowed. So I think most of you are probably familiar with what those two different terms mean, but you can see with the pictures the windrowing, the harvest residues that was retained was essentially pushed up into these larger linear piles of down wood, or harvest residue. Whereas the sheared sites in North Carolina, the distribution was a little bit more even and the piles weren't as large. And again, we'll get at some of the differences between the sites a little bit more in the discussion. So just to give you a basic idea of the layout in terms of what our treatments look like, we had 6 treatments, and again, these are based on differing volumes and distributions of harvest residue. So we have a 15 and 30% retention, each of which are divided into two separate treatments: the clustered distribution and then the dispersed distribution of those harvest residues. Then we have a no biomass harvesting guidelines treatment. In that treatment, essentially all the down wood was removed that would have normally been deemed as woody biomass during a woody biomass harvest by those that are removing it. And then the no biomass harvest is all the harvest residue was retained. And these clear cuts that we're working in are pretty big. They range from 6 to 14 hectares. I think probably on average they're about 8 to 10. So just to get a little bit about the methods here, we're counting birds. We're walking around in clear cuts and we're counting birds. Right? We counted birds in North Carolina only for the winter portion of the study, but then for the breeding season we sampled birds in North Carolina and Georgia. We also sampled vegetative characteristics in the clear cuts, and then in a separate study, for those of you, or in the concurrent study, I should say, my colleague Sarah Fritsch, she quantified the down wood volumes, which we will use in the future, but those aren't presented in this webinar today. So for the winter birds, we sampled from 2012 through 2014, so we have 3 years of data, and all that data has been collected. And our 2014 breeding bird field season is in progress as we speak. The 2012 and 2013 data has already been collected. So we'll have 3 years of data for the birds, both in the winter and the breeding season. Just to give you a little bit more detail in terms of where we were recording birds. So this is a map of Clearcut, one of the sites in North Carolina. The treatments are delineated by the blue lines, and the dots represent different bird detections. So I'd just like to make it clear that what I'm going to be presenting today really pertains to these treatment detections. So these are birds that we're detecting within the treatments, and because we're detecting them within the treatments, and those treatments have certain vegetative characteristics, the downwood is they're most likely to be responding to the treatments in terms of the harvest residue retention in terms of these birds that are detected in the treatments, these treatment detections. So we also recorded birds on the edge of the clear cut. So you can see here in the picture the star is indicating that clear cut edge, and this is sort of the layout in North Carolina where you'll have the clear cut edge, a logging road, and then an adjacent forest stand. So we also recorded birds in those adjacent forest stands. And then lastly, in North Carolina, we recorded birds in the interior ditches of the clear cuts. So the idea here is that they're in the interior of the clear cuts. They are on the edges of treatments, but these ditches surrounding the treatments that are inside the clear cut have different vegetative characteristics. So we grouped all these detections together and then analyzed them separately from group to group. But today we're going to be talking about the treatment detections, right? So these are the birds that are most likely to be responding to the harvest residue retention. So now to bring it out to Georgia, we also have treatment detections in Georgia, right? And then we have adjacent forest edge detections, and we have detections along these wetland retention forests that were sort of scattered throughout the Georgia sites. So we'll be addressing all of these to try to get at some sort of more landscape level response, or at least for the stand to address the surrounding habitat. We're going to be looking at these edge detections, but we're primarily focusing on the treatment detections thus far. For the invertebrates, what we're doing is pitfall trapping, which is a method by which you can collect ground-dwelling arthropods that are sort of scooting along the ground, and then they just fall into a cup. So we have 3 treatments in North Carolina for the invertebrate sampling. There's a hardwood pile, a pine pile, and then no pile, right? And just to give you sort of a layout of the trap arrays in this middle picture here, the clear cuts are sort of uniform in that you'll have a bedded row and then an inner bed and then another bedded row. So the idea is we have one trap on either side of the inner bed in a bedded row, and then 2 traps on either side of the debris that is it's in the inner bed because of that shearing, right? The debris is in the inner bed, and that's where we're trapping invertebrates. Now, of course, for a pitfall trap array that is in a no-pile treatment, the picture in the middle there would be exactly the same, except there wouldn't be that down wood in between the 2 traps in the inner bed. In Georgia, our methods were a little different because, for one thing, most of the debris in Georgia was pine, whereas the debris in North Carolina, in terms of the treatments, was primarily hardwood, and then there was some pine mixed in, so we were able to get at those 2 different types of wood. But for Georgia, we're just really focusing on pine piles, and the piles are essentially windrows, right? Because whatever harvest residue was left in those treatments was then windrowed up into these big, was pushed up into these windrows. So we're sampling invertebrates in Georgia with the same sort of layout in terms of the trap array, but we're sampling around a windrow and then areas with no windrow. And these traps are about, at most, 50 meters away from one another, but they're generally 25 to 50 meters away. So you have that independence in terms of the ground-dwelling arthropods, but you still don't want to be trapping them way away from the other trap array because you might have some microsite characteristics that differ, so we're trying to reduce the chances of that. So again, there's some preliminary results for the winter birds. So just to describe the layout here of these bar charts, they're all going to be the same. In terms of the X and Y axis, right? So on the X axis, we have the 6 treatments that we implemented for our study. And then on the Y axis, we have our response variable, what we're looking at. So it'll either be diversity or abundance, but just be aware, the treatments are on the bottom and the same layout. We're going to be using that same layout. The error bars are standard error. Again, this is just preliminary results. I want to emphasize that, and this is data collected from 4 clearcuts, 2012 to 2014. So you can see here with our first graph, we're looking at an index of diversity, and we standardize per visit as well, just so you know. But mainly, from here on out, we can just look at the pictures. So you can see here that we have diversity decreasing from year to year, which is sort of interesting, and we'll discuss this a little more in a bit. But in terms of the index of diversity for winter birds, we do see a trend, a decreasing trend from year to year. But then when we look at relative abundance, which is this next graph, you can see that the bars in blue, 2012, pretty low relative abundance compared to 2013 and 2014 in the orange and gray respectively. And again, we're going to talk about why that might be in a few more slides. So now, birds, it's sort of an overgeneralization to just look at winter birds' response to these treatments as a whole, because their behaviors and relationships with down wood vary between species. So what we did is we took some of the more common species we were running into in the clearcuts, and also some species that it would make sense that they would respond to down wood, and plotted those out. So here we have Carolina wren. So Carolina wren is a bird that is typically thought of as being at least somewhat associated with down wood. And what we can see here is that we have a definite difference, at least in 2013 in the orange there. It seems that the mean relative abundance of Carolina wrens was actually higher in treatments with more harvest residue retention than those with less, which is indicated by those red circles. So, these data have not been analyzed yet, but so it's difficult to say whether there are significant differences. But just based on a visual assessment, looking at the standard errors, it does seem that there is some trend in that there's higher abundance in that some of those treatments with more downwood retention. Okay, so we have house wrens here for the next slide. And again, what we're seeing is what appears to be a trend in which, similar to the Carolina wrens, in which relative abundance is higher in that no biomass harvest treatment, and then that 30 cluster in 2013, so these are the orange bars, then in those treatments to the left, which are treatments with less downwood retention. So I should also mention that on that x-axis there, the treatments are listed in order of basically the least amount, the lowest volume of downwood, and then as you go to the right along that x-axis, you're getting to treatments with more and more downwood until you get to the no biomass harvest, where you're dealing with a treatment with the most downwood in terms of our experiment. So song sparrows were fairly common in the clear cuts where we were working. Note that this graph is just 2013 and 2012, or sorry, 2013 and 2014. You can see that there's really doesn't appear to be any difference in terms of the mean abundance, relative abundance of song sparrows among treatments. Similarly, we see a limited response by swamp sparrows to the treatments, where there appears to be a year effect. So from 2013 to 2014, there were definitely more. It appears that there were more swamp sparrows, but if you just look at the overall trend of those bars across years, there doesn't appear to be a treatment level response. So just to discuss these these preliminary winter results, what we have here is sort of a successional timeline that we're dealing with. So in 2012, the sites were really just bare ground. Vegetation hadn't necessarily reestablished at that point. In 2013 and 2014, what we see is from the previous year's growing season, sort of an influx of this grassy vegetative cover. We also have relative abundance. So with this green arrow, the relative abundance is increasing through time of winter birds. But we also see that the diversity is decreasing through time. So what appears to be happening is, first of all, in 2012, we have an overall low abundance of birds to begin with. In 2013 and 2014, once we see the establishment of that grassy cover, that's really when the sparrows moved in. And what we saw was, these sparrows, they're really driving the relative abundance, the increase in relative abundance, because there were just tons of sparrows in these clear cuts in 2013 and 2014. Meanwhile, diversity is going down, which may be just a case where, we have issues where we have a few species that are really abundant, and then that's affecting our diversity index. So, it looks like, in terms of the relative abundance of these sparrows, that they're responding to the availability of vegetative cover in the clear cuts more so than the availability of harvest residues or downwood, whereas birds like wrens seem to be, at least marginally or perhaps even to a significant statistical level, responding to treatments at least with more downwood than less, right? So we'll move on now to the breeding bird results. So I broke those up into North Carolina and Georgia because they are two different sites in more ways than one, just beyond geographically. And I'll talk a little bit about that in a second. So again, same layout here with the graphs, treatments on the x-axis, increasing in volume of debris from left to right. And these are just the 2012 and 2013 results, because our 2014 data is coming in as we speak here. So again, looking at diversity, in the case in North Carolina, what we see is sort of something a little bit more of what you would expect to be happening, where you have an increase in this in the index of diversity from 2012 to 2013 as vegetation is growing in these clear cuts. Relative abundance follows the same trend, and it's more exaggerated. So you can see that the relative abundance of breeding birds in these clear cuts did not differ, don't appear to differ between treatments, but there appears to be a year effect where the relative abundance is a lot higher in 2013 than it was in 2012. So again, we looked at some species-specific responses to the treatments. So here is a graph for Carolina wren, and you can see here that we have on this right-hand side of the graph where you have those treatments with more downwood retention, similar to winter. It appears that Carolina wrens are at least slightly more abundant in terms of relative abundance in those treatments with more downwood than those with less. However, when we look at birds where we may not necessarily a priori expect a response in terms of their relationship with downwood, so looking at some of these neotropical migrant passerines like a blue grosbeak, what we see here is that there doesn't appear to be any response of blue grosbeaks to the treatments. So you can see all these bars are more or less in line with one another from year to year, and also there didn't appear to be a year effect from one year to the next, at least for blue grosbeak. And that's the difference between those lighter green bars in 2012 and the darker green bars in 2013. So we have field sparrows. A pretty obvious year effect here. So there were a lot more field sparrows at our sites in 2013 than in 2012. But again, no real treatment level effect is apparent. Again, indigo bunting, same deal as the field sparrows, where you're seeing an increase from one year to the next, but no apparent difference among treatments in terms of the relative abundance of indigo buntings. The common yellowthroat, really, they came in mostly in 2013, so there weren't as many in 2012. In fact, there were so few that we didn't. I didn't even bother to show them here, but this is the 2013 data for common yellowthroats, and interestingly, they seem to be at least slightly more abundant in these treatments with more downwood retention on the right than those with less downwood retention on the left. Moving on to the breeding bird results for Georgia, most of the species-specific responses for birds that I just showed you in North Carolina applied to those in Georgia as well. So I'm just going to show you a few sort of baseline graphs describing the breeding bird community in Georgia as a whole. So this is Index of Diversity, and these data were collected from three clear cuts in Plum Creek and one in Georgia Pacific. So you can see here from 2012 to 2013, we see a decrease in diversity for breeding birds. So sort of interesting, these diversity indices it's debatable how useful they are in some cases for describing what's going on in terms of a treatment level response to an experiment, but it is kind of interesting how we have sort of varying results between the sites and between seasons as well. But then when we look at relative abundance, it appears that birds were similarly abundant in terms of relative abundance between years and then also between treatments. So there doesn't appear to be much of a treatment level effect. So in terms of discussing why this might be, these birds are responding, many of these birds are responding to the sort of revegetation situation, right? So you have, as these clear cuts, the successional process is beginning after these stands are clear cut, and what you have is vegetative structure increasing at an accelerated rate, and then also the harvest residue that is retained. As the stand ages, that downwood is decaying through time. And one study by my advisor, Chris Mormon, has shown that, for the most part, downwood in the southeast is pretty much disintegrated, within 5 years. So there's this sort of inverse relationship between downwood decay and this influx of vegetation. Meanwhile, most of these birds appear to be responding to changes in vegetation in these clear cuts, more so than the retention of downwood, save for a few species that are typically associated with downwood to begin with. So again, here's our this red circle indicates the area in which we're working in terms of a loose timeline. I mean, there's no scale on this thing, but you get the idea. And then site prep, right? So we're talking about the shearing and versus the windrowing. I do think site preparation has something to do with the way birds can respond to, and probably invertebrates respond to these, to woody biomass harvest, because what the site prep does is in terms of shearing, it puts this harvest residue sort of in an evenly distributed low concentration, whereas with the windrows, it puts the harvest residue in a high concentration that's generally, the windrows are pretty far apart at the Georgia site. So, with Georgia, it could be something I'm going to go ahead and coin the dinner in the stomach effect, where, you have a nicely partitioned dinner on your plate with the vegetables and the meat. Maybe it's a fancy restaurant, but then you eat it and it all goes in your stomach and mixes together into one burly mass, and that appears to be what's going on in Georgia in terms of, you have these treatments, but then they're all the harvest residues being pushed up into the windrows anyway as a result of the site prep. So I think some of the take-home messages, and again, this is preliminary stuff, so something to keep that in mind. But with the birds, most of the birds seem to be responding to changes in vegetation in these clear cuts more so than retention of downwood. And in terms of the scale of landscape change, where these birds are deciding to breed, if they're responding to sort of larger scale landscape changes, that seems to be better represented by changes in vegetation than it does by the downwood retention. And the revegetation of these clear cuts is making the downwood less and less available year to year because it's growing over the downwood and the downwood is decaying. Site preparation probably has something to do with how birds respond in terms of differences between North Carolina and Georgia. That's something we're going to look into in more detail. You also have potential edge effects where birds using clear cuts are also going to be responding to vegetation outside the clear cuts, and then also, they could be responding to, well, yeah, we'll just leave it at that. There's some adjacency issues in terms of potential edge effects. So what about the invertebrates, right? Yeah, they take a long time to identify, so I don't really have a whole lot of results for those yet. So I'd like to share with you at least some of the plans we have for the invertebrates. So invertebrates are very effective ecological indicators. So, we're thinking bottom up. So how did the invertebrates respond, and how might that relate up the trophic levels to other animals of interest? And that's really kind of the approach that I'm taking. There are appropriate study organisms to address sort of the smaller scale responses to these harvest residue retention treatments, because if you have animals that are responding to changes on more of a landscape scale, like I mentioned with the vegetation, it's possible that if you sort of zoom in on what's going on in those clear cuts with that downwood retention, which you can do with the invertebrates, you might see a different response. And I'm thinking that that might be the case, at least for some invertebrates. Also, invertebrates are important food sources for basically everything. and they tend to stay put, so you don't really have to deal with these birds are flying around and whatnot. Whereas if you catch a ground-dwelling arthropod in a certain area, you could probably say with more confidence that it's definitely using that area potentially exclusively, except for these they might move around and large-scale movements every now and then. And then also, many of these groups of invertebrates are closely linked to downwood, and that's been well established in the literature. So I'm interested to see, how they respond. So we're taking a multi-pronged approach to this invertebrate work. We're going to look at the invertebrate community as a whole, so trying to identify everything down to family, and look at the invertebrate community as a whole, and then break it out into these bioindicator groups and look at those groups in more detail, so identifying those to genus or species. So the three groups that we sort of keyed in on based on the ones that we're finding and what would make sense to use would be ants, and then ground beetles, and then orthopterans, so crickets mainly. The ant stuff could be cool. Ants are cool in general, but also it might be interesting to see, there's tons of fire ants out in these clear cuts, and they appear to be sort of homogeneously distributed throughout the clear cuts, but I'm interested to see, how the interaction between downwood retention, fire ants, and other ants and the ant community as a whole. So we'll see where this goes, but I'm definitely excited about it. And I think it's going to yield some interesting results that we may not have seen if we didn't look at the invertebrate community. So just to reiterate, these results are preliminary. I'd be happy to come back a year from now and share some results that might be a little bit more, that I'd be a little bit more comfortable with, as I work through some of these data. With that, thank you. Appreciate your time. If anyone has any questions, my email address is up there in the right-hand corner. Also, a little plug for the biomass, the best team here. It's a team of biomass ecology folks, and the website is listed there. So if you guys want to get some more information, you can contact me directly or check out our website, or stay tuned for some more finalized results in the future. And I'll take any questions. Thank you.
Okay, thank you, Steve. We have had some questions that have been submitted throughout the presentation. Brian has had several questions. The first was diversity index. Was it just birds, or was it birds plus arthropods?
Yeah, so the diversity index, it was just birds. So we don't have any, I didn't present any results for the invertebrates yet because we're still working on identification and whatnot. So the diversity index, I think I presented 3 of those graphs. I presented a diversity index for the winter birds, the breeding birds in North Carolina, and then the breeding birds in Georgia, and those were just birds.
Okay, thank you. He also had a question. Have you looked into species diversity and abundance for small mammals and reptiles for these treatments as well? And you can comment about that, and then I'll comment.
Yeah, so I guess so that has been done, not by me, but by my colleague Sarah Fritz, and that webinar was presented. That was the webinar that was done before this one, so I believe it's archived, and information pertaining to the small mammal and herpetofauna response can be found in that webinar, and then also some publications and some extension work that will be coming out as a result of Sarah's work.
Right, good point. It's archived, and also on that website that Steve put up for our team, Sarah has several presentations that are there that you can look at as well. Damien asked the question, are any of these birds protected, threatened, or declining generally in these areas? Right.
And that's a good question. So what I'm hoping to do in the future is potentially incorporate there are these partners in flight sort of scores given to birds based on conservation concerns for individual species. So that's a sort of a quantitative way to look at bird response in a conservation context, but in general, most of the birds that are using these clear cuts are fairly common. A lot of them are generalists, and there's not really there aren't really any species that are of very serious conservation concern. There are a few that arguably could be considered, at least partially. I don't know what the term would be, but none of them are really listed in any official way.
Okay, thank you. Ron's asked a question. Have you noticed a decline in avian populations during a biomass harvest during nesting season?
Yeah, so for the breeding bird work that we were doing, we were sampling birds beginning with territory establishment and then all the way through to the end of the breeding season. So we were sampling birds during a time at which they were nesting. Now, we didn't actually go in and do things like nest searches, but what we are doing is we're going to be able to create territory maps for these birds and then relate that to the treatments. So sort of those territory maps are at least partially representative of breeding bird response for birds that are nesting, because if they're maintaining territories, it's pretty likely that they're nesting. It's just that we don't know, their response in terms of when they're actually nesting versus when they're establishing the territory versus the when the fledglings are out, that fledging period. So what we have is sort of a broad assessment of breeding bird response, which incorporates the period of time when they are nesting, because we collected those data during the breeding season in Georgia and North Carolina.
Okay, thank you, Steve. Would point out Robert Barden has again put up on the chat window the website for the overall project, and there are a number of presentations and publications there. David has asked a question: who is looking at overall site productivity, that is site index, over time? We are not doing that as part of our the wildlife group. The folks at University of Georgia, several folks there, including Larry Morris, are looking at soil characteristics. And Bob Barden, has that presentation been done yet? I know they're doing one of these webinars, and I can't remember whether that's been done or not.
So I don't believe it's been done yet, Dennis.
So stay tuned for that one, although it's not specifically looking at site index over time. There has been some of that work done in the literature. Dr. Lee Allen of our department here has done some of that you can do Google search and get some of that information there. I know that's a quick and short answer. And Bob, I think we're running short of time, so any instructions?
So what I'm going to do is I will push out the evaluation for people to take, and those that are interested in getting continuing education credits will need to complete the satisfaction survey, take and pass the short quiz, and then complete the form at the end of that so you can be emailed your certificate. Once you've completed all three parts, your certificate will be emailed to you directly upon completion of those three things. And I'll also publish it here in the chat window for those that are looking for continuing education credits, and for some reason your browser doesn't launch with the form. So I'll publish it here, and like I said, in the chat window. Hold off on clicking on that because I'm going to also push it out. But if for some reason when I push it out it doesn't open your browser in a minute or so, you can always click on that link and that will do it. Remember, there's approximately 50 of us online, and so it's going to be launching all 50 of us at once, so it does take a short period of time for it to launch. And with that, Dennis, we can open it up for if there's any more questions that people have that need to be answered.